give you, this is one of my most precious parenting secrets, okay? I make a deal with them every year. When they collect gigantic You're welcome. Not enough trick-or-treating yesterday. I totally get it. When they come home with massive piles of candy, we have a deal. And that deal is you can trade in some of your candy for, other, even more precious things like toys, D&D figures, Magic the Gathering cards, So then they're happy to share with you all the fruits of their labor. Okay, I'm just kidding. I'm actually not kidding about the trade that we have, but usually my mutation lecture is somewhere right around Halloween, and when I'm lucky, it falls right on Halloween, and that's an awesome thing because it just seems appropriate, a lecture about mutation and its effects on Halloween. All right, this year it's the day after, but it's still a great lecture. Okay, before I start, just a couple of things. One is I have already, and this year it's super early, I've already had requests to post old exams, and I will post an old exam for you, but I want to explain that, as I have emphasized right from day one, this part, this section, the second half of the course is very different from the first half. In the first half, you all practiced problems until you were experts at them. You knew exactly which problems were going to be on the exam. You had practiced them many times, and then you would see versions of those problems with the numbers, different, with minor variation on the exam. Now, as I said, this part, this half of the course, is honestly not really about problem solving, right? We get to do the fun stuff. We get to do the conceptual understanding. So it's really, with maybe just a few exceptions, really not about problem solving. It's not about complex pedigrees, right? If there's any problem solving, and I'm certainly happy to go through the very few, very simple problems that there might be, but it's really about concepts. So I am going to post an old exam to give you a sense of the types of questions. I'm the one that writes all of the exam questions, and I've done it for years. So I'll give you a sense of the types of questions I like to ask. However, it's very different from the first half, because if you see a question on the practice exam, that's not going to be on the final, right? Because it has to be different. I can't give you the same questions over and over, because it's not like, with the few exceptions of the problems, it's not the same as lots of problem solving. On the exam, I'm going to ask you about concepts, your understanding of the concepts that I've presented, and ask you to reason through those concepts, see if you actually understand them, okay? I will talk about it in a little bit more detail in the second hour, but that's pretty much how it's going to go. All right, just the second thing before I start, and that is, I am going to give you a little, talk a little bit more about maybe some of the types of research and questions that we're interested in my lab today and in the future. We are looking for highly motivated, enterprising, young undergraduates. I love to have undergraduates in my lab. Undergraduates have always added a really dynamic, enthusiastic component to my research program, and the best and the brightest have even been involved in research papers and going to international conferences. So if you think you might be one of these undergraduates, please let me know. We are specifically looking for undergraduates with computer expertise. If you have bioinformatics expertise, okay? So if you happen to be one of those undergraduates sitting in my HMB class, and I totally understand it's not going to be everyone, but traditionally we have gotten amazing undergraduates from this class. So if you have bioinformatics expertise, if you're a bioinformatics specialist, if you have familiarity with Linux systems, please send me your CV and a statement of interest, okay? All right. Okay, so today, we are now entering into the really fun part of the course. This is where we get to talk about underlying mechanisms and applications, right? We're at a really critical moment in biology. We have had game-changing advances in methodologies, right? And really, you're the generation that is going to be the one to understand how this all plays out. So, this is where, but you need to understand the underlying biology and genetics. Okay. All right, so our focus on this next section of the course will be on understanding underlying mechanisms, so we need to start at the beginning, all right? We're going to start small with mutations, and just to make sure we're all on the same page in terms of terminology, all right? Now, you might ask me, do I need to memorize all these terms? Like, what is actually important? You tell me I need to understand about underlying concepts. How is it going to benefit me to memorize terms? Well, the issue is, and this is true for scientists as well, for research scientists, how are you going to know what people are talking about if you don't know what the terms mean? For scientists, it's all about precise definition and usage of terms. And as you could probably see from the public discourse about science, a lot of the misunderstandings and miscommunications arise out of people using terms in different ways and meaning different things. That's why scientists are very precise in their language, okay? And that's why I want us to all start off on the same page. Okay, as I said, usually this lecture is somewhere around Halloween, and so this slide of animals that resulted specifically from the Chernobyl nuclear disaster that happened in 1986. These are real pictures, by the way. They are not photoshopped images. And of course, this was one of the worst nuclear accidents in history that is still having effects today. Okay, one of life's ironies, of course, is that more recently, the absence of humans from this area that was cordoned off, it's actually become a haven for wildlife despite the high levels of radiation decades later. There's still an exclusion zone of almost 1,600 square miles. Okay, so let's talk about mutation. The term mutation covers a lot of different types of genetic changes, everything from changing a single base pair to, large stretches of nucleotides, all right? Today, we're covering specifically mutational events that occur within single genes. Future lectures will cover larger scale rearrangements and losses and additions. Okay, so one of the really interesting things about mutation is that it sounds like an artificial thing. It sounds artificial and bad, right? But in fact, a lot of mutations simply arise spontaneously due to errors in replication, all right? So due to the natural error rate of polymerase enzymes that have to replicate your entire genome, right? For every cell, there are introduced changes that happen that we call mutations. Now, of course, this is not the only way. These random incorporation errors that happen is as a result of the natural processes of DNA replication. That's not the only way that mutations can occur, of course. There's also the more colloquial meaning of mutation, which is a result of exposure to environmental effects, whether it's radiation or chemicals in the environment that result in changes in the bases of your genome, okay? So the process of mutation, however, whatever is the root cause of it, it's the process by which genes change, right? But the process by which they evolve new alleles, right? It occurs randomly, it tends to occur randomly, but also can arise spontaneously, not just during normal replication, but by the action of a mutagen. Of course, I should remind you, you guys probably all know this already, but only mutations in germline cells are transmitted to progeny. Somatic mutations can actually occur at an even higher rate, but they only affect the cells in which they occur, okay? And then inherited mutations, you might hear them referred to as alleles. New alleles that arise in populations of individuals of a species, and when they arise, they occur at very low frequencies to begin with, right? And then they might stay at low frequencies, they might disappear, or they might be selected for. Okay, so, all right. General observations of mutation rates. Mutations, especially those affecting phenotype, but mutations in general, occur very rarely. Different genes tend to mutate at different rates. The rate of forward mutation, right, the rate of acquisition of new mutations is almost always higher than the rate of reverse mutations back to wild type. As I mentioned, mutations can occur during normal DNA replication, and the mutation rate can increase after exposure to a mutagen, right? Whether it's radiation or chemicals, the overall effect is actually to increase the rate of mutation, increase the number of new mutations that are happening. Now, we can talk about classifying mutations into different types, depending on exactly what they are. Of course, you all probably know that the wild type is the starting sequence. That's deceptively simple. It's actually the allele that's the most frequent or predominant, okay? Substitution is when a base is replaced by one of the other three bases. A deletion is when you lose one or more nucleotides, versus an insertion is when one or more nucleotide bases are added, right? We often call these two indels, and the reason is because we often can't tell the direction in which it occurs. It might sometimes be difficult to discern, especially if you compare between different species. One species has a nucleotide there, maybe it was an insertion, but the other species doesn't have nucleotides in that spot, but maybe it was a deletion. And so in order to figure it out, you need external information, right? So we tend to call those indels so we don't have to always determine in every case whether it was an insertion or a deletion. All right, inversions are a segment of DNA cut out, inverted, right, and put back in. Reciprocal translocations, you're going to hear about it more in a minute, are when parts of two, not in a minute but in future lectures, parts of two non-homologous chromosomes get cut off and change places and stuck back on onto chromosomes that are not homologous. All right? Okay. So classification of mutations, you can have base substitutions, right? You can have transitions in which, of course, everybody remembers that there are two different types of bases, purines and pyrimidines, and they have very different structures. And if you have a mutation or a substitution in which you substitute from a purine to a purine or a pyrimidine to a pyrimidine, so in other words, you don't change the type of base, that's called a transition. And that is much more frequent. It's a much more frequent type of mutation in almost all cases than transversions, in which you change the type of base in the substitution, okay? All right. So why study mutations? There are many, of course, many different reasons to study mutations. The most obvious being that mutations can be associated with disrupting function and can affect phenotype. This is not always true, right? You can have mutations in non-coding regions. You can have mutations that are synonymous mutations that don't affect the amino acid that's coded. You can have lots of different types of mutations that are not actually disrupting gene function, right? But for those that do, they're obviously very interesting to study. The other reason for studying mutations, of course, is something that I've alluded to in the previous lectures, and that is that these mutations, which in humans are called SNPs, or single nucleotide polymorphisms, can act as markers for studying the effects of loci, linked loci, which are associated with interesting phenotypes that we might want to know about, okay? So for these two reasons, and actually you could probably think of many more as well, it's very interesting to study mutations. And we know more and more about them. Okay, so definitions, more definitions of mutations. The wild type is the form that's found predominantly in nature, all right, or in a standard laboratory stock. And it usually, biology is all about rule of thumb. It's usually it's usually the majority, all right? So it has to be an allele whose frequency is at pretty high levels, relative to a mutant where the frequency is found at very low levels, because it's usually something that arose pretty recently, and so it's usually not more than maybe 1% of the population, okay? A forward mutation, as I alluded to before, changes from the wild type to a different mutant allele. Whether that mutation is recessive or dominant, doesn't matter, right? It's still a forward mutation. And a reverse mutation is one that causes this novel mutation to revert back to wild type. So in these two examples here, right, we have the forward mutation to a mutant allele, and then the reverse mutation that reverts back to wild type. Okay, causes of mutations. As I said, they can arise through the normal processes of DNA replication in the absence of any known mutagen, simply because there are so many base pairs that need to be replicated, right? Billions of base pairs that need to be replicated. Even the best possible DNA polymerases that we have a certain error rate, right? And because they have an error rate, even if they're fantastic, they've got lots of, proofreading activities, they still have a certain background rate of mutation. That's it's just inherent in the process by which replication happens, right? So this gives rise to a background rate of mutation that can, of course, vary in different animals, depending on what you're talking about. It varies in terms of, nuclear versus mitochondrial genes. It varies in bacteria that doesn't have as much in the way of proofreading as, say, a human or vertebrate DNA polymerase does. But in general, we think about the background rate of mutation as somewhere between 2 and 12 times 10 to the minus 6 mutations per gene per gamete, okay? Now, this is just the spontaneous rate of mutation. It's actually pretty low, if you think about it. But this can be greatly raised by the action of mutagens that act to elevate the rate of mutation, all right? And so it becomes something that geneticists can use as a tool in the laboratory in order to elevate the rate of random mutations that are happening so that they can be studied, so that their effects on phenotype can be studied in the laboratory. All right? Okay, so let's talk a little bit about these. What causes spontaneous mutations? There's depurination, which is the hydrolysis of a purine base. Purine bases are these double ring ones, so they are more susceptible to hydrolysis, right, than other types of bases. And it's the hydrolysis of a purine base from the deoxyribose phosphate backbone, all right? This is thought to occur approximately 1,000 times an hour in every human cell. And when this happens, of course, then this, destroys the information content in the nucleotide, right? So the apurinic site does not specify a complementary base, and the DNA replication process has to try and fix that. And when it does, it occasionally introduces a random base opposite the apurinic site that causes a mutation in the newly synthesized complementary strand, okay? All right, so we also have deamination, all right? And the problem with deamination, which can happen to cytosine, is that getting rid of this amino group means you actually get another base. You get uracil. So you have deamination where the cytosine is converted to a uracil, but a uracil will actually base pair with a different base from the original cytosine. So when replication happens, what happens is that on this strand, an A is synthesized, whereas on this strand, a G is synthesized, and you have a mutation, okay? All right. Again, it's the loss of the original information that was contained. All right, so what can cause induced mutations? One of the forms of radiation, as you might know, that's the most pernicious to double-stranded DNA are X-rays. And X-rays are particularly bad because they can introduce double-stranded breaks in the DNA sugar phosphate backbone, right? There are other, types of radiation that can also do this. Other types of ionizing radiation like gamma rays, certain types of radioactivity. But the fact of the matter is, when you have a double-stranded break like that, you have some possibility that that entire segment is going to be lost, is going to be deleted, right? These two could be rejoined, and then you basically lost that intervening segment of DNA. All right. There's also the effects of UV light. High energy UV light can form, if you have adjacent thymines, they will form thymine dimers. They're then detected as errors, excised, and when it's repaired again, you can have substitutions that happen in its place that are incorrect, all right? Other types of damage that occur can be oxidation. Oxidative damage, it can happen to any of the 4 bases, and they also can result in changes in alterations of the information content contained in the bases, as you see here. So oxidation of a guanine can lead to alternative mispairings with other bases, such as the A here, which is not what the original was supposed to be a C, okay? And then with subsequent rounds of replication, those errors are propagated. Okay, you can also have indel mutations, and these are, again, these can arise spontaneously from the replication process itself, and these tend to arise particularly frequently in regions of repeated bases, such as this stretch of T's here. Because as you can see, it's very easy to have a loop form and mispairing form during the process of replication, right? Where then you have either an addition or deletion, in this case an addition if the loop happens on this strand, right? Or a deletion if the loop happens on this strand. All right? So often this happens because these loops are actually stabilized during DNA replication. Now, some of the molecular consequences of mutations in coding sequences, in protein coding gene sequences. These types of mutations, these types of point mutations, can result in synonymous mutations, and those are ones that don't actually change the amino acid that the codon is coding for because they're silent. For example, a lot of third base position mutations are like that. You can also have non-synonymous or missense point mutations where the point mutation results in a change in the amino acid that it's coded for, all right? These types of non-synonymous or missense point mutations that simply change the nature of the amino acid don't change the size, of the RNA transcript as measured on a northern. They don't tend to change the size of the protein as measured on a western blot. Okay? It's just an amino acid substitution. All right. Now, one of the interesting things that you can do with non-synonymous and synonymous rates of mutation is that they allow for estimates of the strength of selection. Because you would expect, right, synonymous mutations or silent mutations not, in general, to be the targets to be the targets of selection, right? So you would expect them to be effectively neutral, whereas the rate of non-synonymous mutations would be the targets of selection, whether it's negative selection for amino acid mutations that interfere with the function of the protein, or positive selection, those that would lead to an adaptive function, okay? So we can also talk about neutrally evolving genes having similar rates. All right. Now, missense mutations or non-synonymous mutations can differ in severity depending on what amino acid is being mutated to, right? If the mutation is to a very similar amino acid, we would call that a conservative substitution, right? Because we consider it less likely to alter the structure or function of the protein. Now, of course, that really depends on context. If it's an incredibly important part of the protein, for example, the binding site of an enzyme, you have even a very conservative mutation might radically alter the function of the protein. But overall, in general, we can look at amino acid substitutions and group them into different categories, all right? So there's conservative, and then there's non-conservative amino acid substitutions or non-conservative missense mutations. And non-conservative ones are those in which you change to a very different type of amino acid. It's got different physiochemical properties, or biophysical properties of the side chain, so therefore much more likely to alter the structure or function of the protein. You change from a charged residue to a hydrophobic residue, or to a polar residue, right? Or you change the size of the amino acid side chain tremendously. Okay, again, there are other types of mutations as well, which are nonsense mutations. Nonsense mutations tend to have some of the most severe effects of point mutations, and that's because they result in a stop codon being incorporated into a protein coding sequence. This stop codon does not tend to affect the RNA transcript, as you can see on the northern, but it does affect the size of the protein, right? Because it's a stop codon leading to a truncated protein, as you can see here. All right. And then finally, you can also have frame shifting mutations. So insertions or deletions, in general, have a high probability of leading to frame shift mutations, as you can see here. So the original sequence, wild-type sequence, look like this, but if you have a frame shifting insertion of a single base here, it totally changes what the downstream reading frame is and what the downstream amino acids are in this protein. And so as you can see, this type of insertion doesn't really affect, it's one base, right? So it doesn't really affect the RNA transcript size, but it can affect the size of the protein, because frame shifting is likely to result in the introduction in the stop codon being read in a place and time that's different from the wild type protein. So it can go in either direction, bigger or smaller. All right. We can also have what we call intragenic suppressor mutations. So if initially you have a frame shifting mutation, so for example, if I deleted this C, right, then instead of the fat cat ate the big rat, you would have this, where it's not a legible sentence anymore, right? Because all of a sudden the frame shift leads to gobbledygook. But on the other hand, you can also have the introduction of an intragenic suppressor mutation that recovers the correct reading frame. So in this case, if you insert the A here, you recover the reading frame for the rest of this DNA sentence that I've constructed here. Okay? All right. Mutations, I'm moving along here because we've got a lot to cover. Mutations outside of the coding sequence, right? So we can have splice donor acceptor site mutations. So it basically are mutations in the consensus sequences that direct where introns are spliced out, right? During the process of RNA processing. To produce mature mRNA transcripts. And if you have mutations, it can lead to incorrect retention or excision of introns that then lead to huge changes in coding sequences. So like, for example, this can lead to the creation of a novel 5' splice site that then actually eliminates part of the exon, right? And so you would have a much shorter exon here than you're supposed to, which would tend to change the reading frame. Similarly here, if you have a mutation here that eliminates this 5' splice site, this intron will no longer be spliced out, and instead the coding sequence that's read by the ribosome will include this intron. Again, leading to the insertion of amino acids that shouldn't be there, and probably leading to a frame shift as well. Okay. Now we can talk about mutations from the point of view of their effects as well. I was talking from a mechanistic basis as to the how to defining exactly the types of mutations based on how they're created. But they can also be defined based on the effects that they have, all right? So you can have loss of function mutations. Those are mutations such as diagrammed here, where you have they're null or amorphic mutations if they are similar to the effects of entirely deleting that allele, okay? That gene, right? So this mutant phenotype, the loss of gene activity in this gene M is exactly matched by the deletion, right? The deletion in a wild-type homozygote. Now, you can also have hypomorphic or leaky expression, right? This leaky expression of the gene where you have this point mutation, but it still results not in a total loss of activity, but it results in some leaky expression that's not as much as the wild-type allele, and that would be called hypomorphic. And of course, these are always in comparison to the homozygous wild type. All right, so some of the consequences of loss of function alleles, mutations are recessive, right? Mutations of haplosufficient genes tend to be recessive. And that's where, when you have only one copy of the wild type allele, in this case, the example is eye color. So one copy of a wild type allele is enough to generate wild type eye color. So as you can see here, the threshold for the amount of this eye protein you need is here. You can cross that threshold if you have two copies of the wild type allele, but you can also do it if you have only one copy of the wild type allele and one mutant allele, right? And here as well. But if you have two copies of the mutant allele, then you do not have wild type eye color, because you're below that threshold, all right? If we are in a haploinsufficient situation, then only one copy of the wild type allele is not enough for a normal phenotype. So the analogy in this case would be that the threshold for a wild type eye color is here. So you have to have two wild type alleles in order to have wild type eye color. Okay? One allele is not enough. And then you can, of course, have gain of function mutations, where you have a mutation that leads to excess protein produced in comparison to the wild type allele, and that causes an abnormal phenotype. You can also, that's a hypermorphic mutation. You can also have neomorphic mutations, where proteins are produced with either a new function or normal protein is produced at an inappropriate time or place, okay? All right, so now I just want to talk a little bit about disease mutations involved in vision, as a subject that's near and dear to our hearts. So here you have light that enters the eye, focused on the back of the eye where the retinal layer occurs. The photoreceptor cells within the retina are what responds to light and triggers the sensation that light has been perceived. The molecules within the outer segments of the photoreceptor cells that actually absorbs light and transduces that signal into a neural signal is called rhodopsin, okay? I'm going to talk about rhodopsin in a little bit more detail here in humans. So we know that in humans we have 4 different visual pigment genes that all react in response to light. We have rhodopsin, we have the SWS, short wavelength sensitive, and then 2 forms of long wavelength sensitive, the green sensitive and the red sensitive opsin genes. These ones operate during the day, right? Red, green, and blue. This one operates in rod photoreceptors at night. That is the whole reason for our trichromatic color system in humans. Now, part of the interesting things about visual pigments or opsin genes is that the red and the green opsin genes occur on the X chromosome. And they occur in tandem like this with one red and possibly a variable number of green genes, okay? This is just a diagram of how the opsin genes are related to each other. Now, it's a fascinating thing that in fact, not only are these red and green opsin genes occurring on the X chromosome, and they occur in tandem like that, but because they occur in tandem, there is and they're very similar to each other, there are many opportunities for mispairing and unequal crossing over events during recombination, okay? And this is just some of the different ways that you can have mispairing events, unequal crossing over events during recombination. So these give rise to a giant amount of natural variation in human red-green vision, including the fact that the rates of red-green color blindness are much higher in males versus females. Because males, of course, have one copy of the X chromosome, and so if that copy has a deletion like this, you will have red-green color blindness, as opposed to females who have two versions of the X chromosome. So not only are the rates of red-green color blindness higher in males, but what this means is that color perception, right? There is a huge variation in humans in terms of color perception because of the effects of unequal crossing over in both males and females. So that is just your interesting vision fact for the day. Okay, in the last few minutes, I'd like to talk about mutations in metabolic and biosynthetic pathways, all right? And experiments to understand the connection between genotype and phenotype at the level of individual mutations in metabolic and biosynthetic pathways. So this is just a diagram. This is just an example of one of those pathways. It's actually a very important pathway associated with many different diseases, okay? And that is the phenylalanine pathway. And this is one of the most famous examples of a disease called alkaptonuria. It was discovered a long time ago in 1902 and by a physician that showed that this is a genetic disorder that's a mutation in a single gene. It's the result of a recessive mutation in an autosomal gene. In this disease, individuals one of the most, the biggest symptoms of this disease is that you have urine that turns black when it hits air, all right? And it's the result of a mutation in an enzyme in the phenylalanine pathway called HA oxidase, okay? And this symptom is the result of accumulations in this compound, homogentisic acid, as a result of it not being able to be metabolized due to a mutation in this HA oxidase enzyme. So individuals can't get rid of this compound. It gets excreted in the urine and turns it black. So it basically stops the pathway. So this physician postulated that they lack this enzyme and that he called this an inborn error of metabolism, reflecting the fact that it's due to a genetic mutation, all right? In this case, a single allele of the enzyme is enough to prevent accumulation of HA, and so that's why the mutant allele is recessive. Now, as I said, a host of other diseases is also associated with the phenylalanine metabolic pathway, not just this one as I showed you in the last slide. There's also the mutation associated with PKU that results in, this is a result of a mutation in the enzyme phenylalanine hydroxylase that results in an inability to convert phenylalanine to tyrosine and results in the accumulation of a toxic compound resulting in the disease phenylketonuria. Okay? So the question is, given the existence of metabolic and biosynthetic pathways like this, where it's basically a chain of enzymes, if you are a scientist, how do you figure out what the order of involvement of these enzymes is? Okay? So this is the diagram of the model that we have in our heads for these, metabolic or biosynthetic pathways, right? It's usually a chain of steps where each is dependent on the previous step. But let's say you didn't know the exact order of the steps, only that these genes code for enzymes that you think are involved in this pathway. So how would you conduct experiments to figure it out? What you would do if you're a geneticist, of course, is that the first thing you would do is knock out, if you think a gene is involved in a particular pathway, you would knock it out, right? So you knock it out, that knocks out the enzyme at a certain step, and it means that now this compound cannot be metabolized to the end, right? So let's say that our assay is a growth assay, and that you knock out that gene and the organism can no longer synthesize the compounds needed for growth or metabolize compounds. All right? So what you can, do is that you can rescue the phenotype. Again, we're thinking like a geneticist. So if we knock this out, and it will not grow, but if then you supply it back the necessary compound, then the mutant organism will grow, okay? So we do that in a series of experiments where we knock this out, we supply back the compounds that we think are in this pathway, and we see if they recover the phenotype or not. And we do that for each one of these individual genes in this pathway, all right? And so we have a nice table of mutants and then the compounds that are added back. And you can, from this data, figure out what is the order that these genes are used and what is the order of the compounds using a few simple rules. That compounds that are used latest in the pathway will support the growth of the most mutants, okay? Compounds that, so for example, G should be used very late because you add it, it supports the growth of all of the mutants. Compounds used earliest in the pathway will support the growth of the fewest mutants. So the fewest would be E. So we can infer from this data that E is very early and G is very late, and so on, right? So E is at the beginning, G is at the end, and so on. So then A would be the compound that supports the next fewest, right? It only supports this one, but none of the rest, and so on, all right? And then you can also take a look at the order of the genes in terms of which steps they must be associated with, right? So E must be the first in the pathway, as adding it supports the least amount of growth. But on the other hand, if you look at it, right, this mutant, right, adding A will rescue the mutant, right? So that means that it must be this first step here, right? Okay. So from that, you can deduce what the order of the genes that are involved in the biosynthetic pathway must be. All right. Thank you all. I will take a 10-minute break, and then those of you who are interested in asking questions in the second hour, you're welcome to come back. So 10 minutes. I'm okay. All right. Okay, so first of all, I just want to thank you all for staying. And I want to particularly thank the people on the discussion board that have been helping to answer questions. You guys have been doing a fantastic job. I wanted to, first of all, just clarify some stuff. I mean, every year it comes up in some form or another. I want to clarify the relationship between the tutorials and the lectures and about the readings. I just want to reassure people that if I assign a number of pages like this, you know, it's clearly, you could probably discern what my intent is. My intent is that you read this section, right? When I say something like this, I mean read this section. So if there's stuff before section 2.25.2 on page 757, I'm not intending that you read that. I'm intending you read the part on mapping QTLs that is, you know, related to what I'm talking to you guys about, all right? So just to reassure you. Now, there was a whole bunch of questions on the discussion board about assign problems for tutorial 6, you know, whether those questions are going to be on the exam, they're not in the readings. First of all, it's two separate, entirely separate parts of the course. I am involved in the final exam. I write the questions for my section of the final exam, and that is completely separate from the tutorial, okay? So things that are assigned to the tutorial, I'm not involved in that. You should ask your TA if you're having problems with questions assigned in the tutorial, okay? Those topics, if they're not assigned by me, they're not going to be on the exam. So questions about how to calculate, you know, GWAS or linkage equilibrium or whatever. It's not, has nothing to do with me. It's not on my part of the final. All right? Is that clear? Okay. All right. Now, I want to go through a whole, there's been a lot of stuff on the discussion board. I do think that the previous lecture was a little bit more complex in terms of its concepts that I was presenting. So I just want to take it a little bit more slowly and make sure that everything is absolutely clear for you guys. So I'm going to show, none of these slides are new. They're going to be slides from my lecture, okay? And as I go along, I'm going to try and address some of the questions that came up that are related to these slides. So first of all, when we're talking about trying to find QTLs, all right? Trying to find QTLs by doing crosses. This is a classic approach in genetics, all right? And it was talking about crossing two inbred lines, as you can see here, two inbred lines that differ dramatically in terms of phenotype, okay? So differ dramatically. Here's one inbred line that's large. Here's another inbred line that's small. And we were talking about tomato size. Okay. So the first question was, this is the cross as I presented it. Two inbred lines, you get F1s. You cross the F1s, and then you study the phenotype, the correlation between phenotype and genotype in the F2s, all right? And I did mention during my lecture that there are many different experimental designs that you can have. This is just one of them. There's some advantages to it. But there are definitely other ways of doing it. And so there was a question that came up that, in fact, the textbook uses back crosses, so crossing the F1 with one of the two parental strains. That is another way of going about it, okay? Here, we're looking at the F2, so I just thought I'd say a little bit about this experimental design and comparing it with the one presented in your book. So looking at the F2 has a number of advantages relative to a back cross. You would get trait estimates for both types of homozygotes as well as heterozygotes, right? Because in your F2, you will see both homozygous as well as heterozygous across different loci, right? Whereas if you do back crossing, depending on which parent you back cross with, you will eliminate some of that variation from that type of homozygote. You know, this is just bonus. I'm answering this question. It's not something that we address directly, but it comes out of, if you think about it a little bit, it's kind of interesting to think about. Okay. The other question was, do the traits have to be additive? And what happens if they're not additive, right? And for these QTLs. Can anybody tell me, are we, looking for additive traits or not additive traits? Or loci, I mean. Additive loci. Yeah? Yes. That was the basis of a lot of one of my previous lectures, that is our model, that we have additive effects across loci, because if we don't have additive effects, what happens? Then the genetic context matters, right? And then it's really hard for us to do this kind of study, because the effects of an allele could be masked. And if it's masked in certain contexts, it's not going to correlate. So that would be really not very good. Okay? So our model is we are looking for additive, QTLs that have additive effects across loci. There could be other ones, but if they are not, we will not see the effects if they are masked by dominance effects, by epistatic effects, etc. Okay? All right. So then I had a lot of questions about why, okay, so you have these F2s, and they're all different. Why are they all different in terms of their genotype? Yeah? Both genes play an equal role. There's a very specific answer I'm looking for. Yeah? That's right, recombination. Recombination occurs, actually, is recombination occurring in this generation? Is recombination occurring in the parental generation as well? Yes, it is occurring. Recombination always occurs. It always occurs, but we're not seeing its effects. Why not? Yes, exactly. It's not resulting in any genetic variation because they're homozygous, so all this recombination is happening, but it's still passing down exactly the same alleles, okay? But in this generation, all of a sudden, you're having recombination happening. Recombination acts at random, right? So recombination is happening at random, and so all of these guys are getting slightly different recombination products. So that's why they're different, okay, in the mix of alleles that they're getting. Okay, so a whole bunch of randomly generated products of recombination. There were tons of questions of various types on the discussion board to do with this concept here. Okay, so for fine scale mapping, right, for fine scale mapping, what we want to do is to take all of these different, we sample thousands of plants in the F2, and what we want to do is they have, as you can see, they have a really high degree of heterozygosity because they're the products of random recombination events, right? So at any particular locus, there's a high degree of heterozygosity, right? And so what we want to do is we want to eliminate that heterozygosity, right? Because what we want to do is we want to determine the effects of we want to identify the QTL, or the locus, that is associated, where the allele is associated with phenotype. And so we don't want it to be heterozygous. We're going to see the largest effects if it's homozygous, right? So this heterozygosity, which is in the F2, now for fine scale mapping, in order to be able to narrow it down to a particular locus, we need to get rid of that heterozygosity. And the way that you get rid of heterozygosity for geneticists in any system is inbreeding. That's right, inbreeding. Why does inbreeding, again, this is another question on the discussion board, why does inbreeding get rid of heterozygosity? That is true. Yes, that is true. But on the other hand, that doesn't fully answer my question of why inbreeding results in getting rid of heterozygosity. Again, it's not a full answer to the question. Yes? That's right. It's basically, inbreeding is basically you're mating with as close a relative as you can find. And your close relatives will tend to be more genetically similar to you, right? And so that's the whole reason. If you inbreed at every single step, right, you will tend to eliminate heterozygosity. So basically, we're inbreeding to allow for the generation of genotypes that are homozygous across, as homozygous as possible across all loci. Okay? All right. And that takes a lot of generations. Now, let me see. I think, so this question was why we need that. Why do we need that? Why do we need it to be as homozygous as possible? And the answer is, I think that this is a question about this slide. Okay? Why do we need it to be as homozygous as possible? And so now, when you look at these inbred lines, right? When you look at these inbred lines, each of these is an inbred line. The red means that it is homozygous at this locus for the red allele, right? Which is, in this case, the large parental allele. Okay? If it's yellow, it's homozygous for the yellow allele. All right? Now, why do we need all of these different inbred lines? Yeah? Yes. Because we need enough different recombination events that we break any association with other loci that are linked but not the cause. Is that clear? Okay, repeat it. All right. So if you don't have enough recombination events, so let's say I didn't have line 3. Let's just get rid of it. No line 3. And we don't have line 4, right? We wouldn't have these crossing over events that isolate this as the gene. Other loci that are nearby that are linked would look like they were the ones associated with this phenotype. Does everybody understand that, or have I made it more confusing? You basically need enough recombination events to break the association with nearby linked loci between the genotype and the phenotype, so that all that's left is the one that's actually causing the phenotype. Yes? These are inbred lines that are the result. Okay, let me just go back. Okay. We've created all these inbred lines over here. So these are the inbred lines that I am now measuring. I take, for each inbred line, each one of those, I take a whole number of individuals of that inbred line. I measure what the fruit weight is, and then I average it, and I put that number over here, and then I genotype the members of the inbred line, and because they're inbred, they should be, all the same. They should all be homozygous, and they should have the same alleles across this area, right? And because I have multiple individuals, I can get a pretty good estimate of what the fruit weight is, because I can average out the effects of the environment. Okay? So then for each one of those inbred lines, right, I do that. And I can see where the association between the allele that they have and the phenotype lies, all right? So does everybody see the association? The big fruit, which is the red allele, is about 180 grams. The little fruit, which is the yellow allele, is about 170 grams. Here again we have big fruit, 180 grams, and little fruit, 170 grams. Everybody see that? Yes? Yeah, you can think of it like that. Sure. Yep. Any other questions? Everybody see the association here? Yep. Well, I mean, I find it useful to think of averages because then the estimates are more accurate, right? Genetically identical individuals, you measure a whole set of them, you get a more robust estimate of what the actual fruit weight is. Yes? That's right. That's right. Red is homozygous from the big fruited parent, and yellow is homozygous for the little fruited parent. Yes. Okay. All right. Okay. So then we had this question. Yes? The the inbred lines versus the nearly isogenic lines, same thing. Okay. This is directly off the discussion board. I'm just plopping it up here. Okay, so this was a question about the last slide. I do not understand the significance of using the inbred lines, the isogenic lines. Why can't we just use the parental tomato strains and introduce a single mutation into the parental strain to test its effect? Why do we use those nearly isogenic lines? And that refers to this slide. It is a great question. I decided I would talk about it because I like that question. Does everybody understand what the question is? Right? So in this experiment, one of the large fruited inbred lines was used, right? And I mean, I think it was derived from the F2 generation. So what do people think? Could you have used the parental strains, and why or why not? Yeah? Oh, a question, okay. Yes, absolutely. Well, so the question, I mean, I don't know if the person that asked the question on the discussion board is actually here. Okay. I surmise, or I interpreted the question to be that after you have determined a candidate gene, why not use a parental strain? Okay? Why use an inbred line that's a result of your F2 generation? Is that clear? Okay, so in this experiment, we have a candidate gene. We have a candidate gene where the allele associated with the big fruited phenotype and allele associated with the little fruited phenotype, right? And in this experiment, we took an inbred line with a large fruited phenotype and we used genetic engineering to put in the small fruited allele into the inbred line that has large fruit. And we saw a reduction in fruit size. So the question, I think, correct me if I'm wrong, is why did they use this inbred line as resulting from the F2, assuming that's what they did? Could they have used a parental strain, the parental strain with the large fruit phenotype? Yes. No, it's So my question is, what is the important characteristic of this inbred line that makes this experiment possible? So the thing is that the short answer, does anybody else want to try? Yes? But remember, we're looking for QTLs that are independent of genetic context. I mean, the way the whole experiment is designed, we're looking for genes with only additive effects, so there should be no genetic context. Yes? Oh, another question. That is a high quality answer. Yes, that is absolutely true. Okay, so in fact, the short answer, the simple answer to your question is you could do it. You could do it in the parental strain. Parental strain is another inbred strain, and you're looking for effects on reducing fruit size, right? So in fact, you should see a reduction in fruit size when you do that in the parental strain. But the high quality answer is what you said, and that is that this is not the only QTL. This is not the only QTL. They chose to focus on F2s that had variation just in this area of interest. So that is why they chose to do it in those strains. It's a high quality answer. I don't expect you guys to, to recreate this on the exam, but the short, more simple answer is that yes, you should be able to also do this experiment in the parental strain, and you should still see a reduction in fruit size. But to make it a cleaner experiment, because they're honing in on this one, possible QTL, and I don't know if you remember from my lecture, when I had a zoomed out picture of all of the possible QTL intervals across the chromosomes, there were other ones that they did not follow up on in this particular study. They chose this one because it looked like it had, pretty large effect. So that's why. And then they chose inbred lines from the F2s that had variation in this part of the chromosome that they were interested in. Okay. All right. Everybody good? Okay. I'm going to move on to LOD scores. And that's because I had lots of questions about LOD scores after the previous lecture. I want you just to remember, now this was in the context of that F2 generation, trying to find statistical associations between genotype and phenotype, right? Because especially when you're looking at correlations between, now this is, I'm backing up a second, this is correlations between markers, right? So these are different markers distributed across the genome. We have different markers, could be thousands of markers. The more markers we use, the more frequent we encounter them, the better we can the better we can do our statistical associations to find putative QTL intervals, right? Because you imagine if I only have 10 markers, I have like 2 markers on chromosome 1, 2 markers on chromosome 2, it's going to be pretty hard to find QTLs because my markers are so far apart. The chances that my markers are anywhere near each other are very low. But if I have lots of markers, then I have a better chance, right? The interval between markers will be smaller, and then I have a better chance of finding these peaks, all right? Now, the LOD score is simply a calculation that gives me an idea, right? It's a statistical framework that gives me an idea of how probable is it that a particular marker, particular region, is actually linked to the trait of interest. Now, because we're using markers, we're not expecting the markers, and this was another question on the discussion board, we're not expecting the markers to actually be the right genes, right? Because we've got this entire genome, and we're only assaying like, I don't know, 1,000, 2,000, whatever, markers. The chance we're going to actually hit that gene is super low. But all we're looking to do is be close enough to look linked to the trait of interest, okay? That's what we're doing here. And because we're looking at, is it linked or is it not linked, we need a statistical way of calculating what's the probability that I'm linked. Okay, and that is the odds ratio, or the log of the odds ratio, right? And all this is the statistical probability, right? It's the probability that you will see the data that you see. So that level of correlation, however messy it is, the probability you will see your data, that correlation that looks like that, given that there is an underlying linked QTL. All right? So that is what that numerator means. Divided by the probability you will see that data and that level of correlation, given that there's no QTL. There's no linked QTL. It's just by random chance. Does everybody understand that? That is the key thing, right? We want to know, we want to be able to assess whether or not we will see this data just by random chance, right? That's why it's an odds ratio. All right? Okay. And then, just to make it a nice small number, we take the log base 10 of this odds ratio. All right? Okay. So everybody is with me. Okay. And then what I did is I gave an example of how to calculate a LOD score from a completely different example. The reason why I chose to give this example based on a pedigree is because it's for illustration purposes. LOD scores can be calculated for all kinds of different applications. It's actually quite complicated, can be quite complicated to incorporate all the factors. The simplest way of illustrating how to calculate a LOD score and how it works is by pedigrees, okay? So this was for a particular disease, as illustrated there, right? And this is for this particular pedigree. The pedigree is affected individuals in black, right? Unaffected individuals in white. And then you can see the mother and the father. The mother is affected. The progeny, the son is affected, but the daughter is not. Okay? So it's pretty clear that this T had to come from the father and not from the mother, right? So these are, basically you have, for this pedigree, sorry, I'm going to back up. I said I would go slower, so I'm going to back up. These are the SNP, right? So this is the SNP on the two alleles. And then this is an autosomal dominant. So if you have the disease allele, you will have the disease. Okay? So in terms of the marker, the SNP, which is SNP1, this individual clearly got the T on the SNP from the dad, and the G from the mom, and also got the disease from the mom, right? So from that, we're trying to we surmise that maybe the G is linked to the disease locus, the disease allele, right? That's not that far away, okay? Everybody with me? Okay. So then we have, and in diseases like this, we basically assume that, the other individual, in most cases, the other individual that's not related to the disease, parent is probably wild type, okay? So this other allele here, and also this is autosomal dominant. Okay, so now we have, this, we're wondering whether these two things are linked. This G at SNP1 is linked with the disease locus. Okay, so now we look at the progeny of these two. Now we have genotyped this woman who is marrying into this family, and the woman at the SNP1 has two Gs, okay? So she looks like this. Wild type at the disease locus associated with G. So she's homozygous in this context, both the disease locus and at the SNP1, all right? So their progeny now have this distribution, okay? So we look at this individual here. Inherit is a GG, so inherited one G from the affected parent at the SNP marker locus, and the other G from the unaffected parent. So this individual has the disease allele with a putative link to a G and a wild type allele, putative link to a G. And these three are all, we would infer them to be all the same. Okay? Whereas the unaffected would be the ones that inherited from this parent the other T allele. So this, and this. They all inherited the T allele, right, linked to the wild type disease locus, and then they inherited the G allele from the other parent. So they are unaffected, all right? But then you have this one. This one is G and T, right? So in this case, they must have inherited the T allele from the affected parent, right? They must have inherited the T allele from the affected parent, but in the affected parent, the T allele is linked to the wild type allele for the disease. So, but they inherited the disease. So clearly they did not inherit the wild type allele from the affected parent, right? So they inherited the disease allele. So there must have been a recombination event in that individual. Okay, does everybody understand that? Okay. So if you plot this out, this generation, the progeny is parental, and then recombinant, like that. There are 8 progeny, one of them is recombinant. Okay? All right. So, the question is that we're looking to answer, using our LOD calculation, is this allele, is this SNP. We've drawn it like this, but it could be really far away, right? Is this linked, right? Is this statistically linked in a way that makes it worth following up on? All right, it's a very pragmatic question, actually. All right, so is it linked? This is how we calculate it. The probability of obtaining the observed results, given that the loci are linked, over the probability that you obtain those results, given that the loci are unlinked, okay? And the corollary is that we have to assume a certain recombination frequency, all right? So in terms of the recombination frequency, we're using the observed recombination rate. I mean, it's the best we can do, right? So for this problem, the reserve recombination rate is 1 in 8, right? So the expected frequency would be 1 in 8. So the expected frequency, the probabilities of obtaining a parental, right, allele, a parental allele in progeny is 7/8. So 7 of the progeny have the parental allele, so it's 7/8 times 7/8 times 7/8, 7 times, so to the 7th, times 1 recombinant progeny is 1/8, okay? So that equals 1/20. Now that's the numerator. The denominator is what would be the probability if the two loci are unlinked. There is no association whatsoever. If there's no association whatsoever, it is equally likely that any child will be parental or recombinant. Probability of 1/2, right? So it's 1/2 times 1/2 times 1 up to the 8th. So that's the denominator, right? Given that it's unlinked. So the numerator over the denominator is 1/20th over 1/256, and that's 12.8, okay? And if you take the LOD score, the log base 10 of that, you get around 1. So it doesn't pass the cutoff of 3 for the LOD score. That's the rule of thumb. All right? So the answer for this is no. Now, one of the, let's see. Okay. Everybody with me? I'm going to take one of the questions from the discussion board, and that was, it's a long question, but the gist of the question, actually for the other ones I basically summarized it. I'm not sure why I put this all up there. But basically the question was, why in this question did we only use, all right, where is it? Okay. Why did we only use the progeny in calculating the recombination rate? Could we, right, we use these. Could we have used this as well? Because in the textbook, some of the problems use different generations as well. And the short answer is the assumption of the recombination frequency is only an estimate, and it shouldn't change much. You should use all available information. You could have said it's 1 in 9 instead of 1 in 8, right? But in fact, we have less information for this generation than we do in this generation to say that that was definitely not a recombination event. We assume that it's not, but so you could have used this. It would have not changed the LOD score very much. It would have not raised it all of a sudden above 3, right? So, yes, that's the short answer to that question. All right, let's see here. So that's that question. So these are very short. I think I answered this question already. How can the SNP be tightly linked to the mutation but not the mutation? How can the marker be tightly linked to the mutation but not actually be the mutation? Very easily. It could be right beside it, but not actually be the disease-causing mutation, okay? This one I'm not going to answer. I just wanted to put it up as an example of something that needs more clarification. It's really pretty impossible to answer a question like that. So just please be more specific when you post questions so that it's helpful for everybody. Okay. And then, what does the number of QTLs mean? Okay. So this person was confused by this statement. Inbred populations like the Finns will have a relatively small number of QTLs for a given trait as compared with other populations. That's right. They will have a relatively small number of QTLs. Now that is really interesting statement, right? That's a really interesting statement. Remember that you can only see QTLs if you have genetic variation. So inbred populations will have smaller numbers of QTLs, right? If you're only looking at an inbred population, you're not looking outside or anything relative. QTLs are talking about genetic variation associated with phenotypic variation. You have very little genetic variation, right? So you may also have very little phenotypic variation. That's why they would have fewer QTLs. Does everybody understand that? Okay. It's not that those loci don't affect schizophrenia. It's just that the Finns don't have any genetic variation at those loci that might have been found to affect schizophrenia in other populations. Everybody get that? Okay. It's a great point. Oops. Okay. And then finally, there was a question. This is the last one. All right. There's a question about people really didn't like the fact that heritability calculations are only true for the population in which they were measured. At the time at which they were measured, the environment in which they were measured. So if you measured heritability for Toronto in 2022, it is not necessarily the same for Montreal, or for Vancouver, or for Toronto from 1922. People really don't like that. I'm not sure why this is so deeply disturbing, but they have tried really hard to provide me with counterexamples. And it simply doesn't work. So some of the counterexamples are, what about extreme cases in which you measure in a population that small h squared, which is, narrow sense heritability, is 1. So no effect of environment. It's all about the additive effects of genetic variation. 100%. Okay? Then, can I, generalize from that to other populations, right? Because then I've showed there's no effect of the environment. And the answer is no, you cannot generalize, still in the extreme case. Because you measured it in a time and a place where maybe there was very little environmental effects. And in a population at a different century, at a different time, there might be large environmental effects, and then h squared will be different.